My name is Neil Bowles. I work in the Department of Pediatrics, Division of Cardiology at the University of Utah in Salt Lake City, Utah. I just gave a presentation at the World Congress on Heart Disease on new approaches to find the genetic causes of congenital heart defects. I covered two areas of our study. The first is to use whole exome sequencing in patients who have a significant family history for congenital heart defects. And I described some of our data looking at one particular family who had pleiotropic congenital heart defects with the major phenotype being atrial septal defects. And I described the data where we identified a mutation in myosin heavy chain 6 gene, which segregated in the family, and we felt probably was a significant contributor to the atrial septal defect. I also described our approaches to find genetic defects in patients who describe themselves as sporadic in that they don't have family history. I described that we are able to actually link these individuals to other distantly related family or distantly related individuals through a utility called the Utah Population Database. And I described how we're using high-density SNP data to actually identify shared genomic segments within these distantly related individuals. And that in the future, that we hope that we'll be able to find variations in genes that are present in these individuals, and that we'll also be able to identify additional families who share genomic segments overlapping with these regions. And using these combinations, we'll be able to narrow down the variations that will be present in these individuals and ultimately identify the genetic cause of congenital heart defects in these people.